In this chapter, we are going to study about cytology. As you have learned in the last chapter, cytology is the study of cells. In this chapter, we focus majorly on three things. Number one, the microscope. Number two, the cell itself. And lastly, we will study one thing about the living thing, the levels of organization, the biological levels of organization, which is quite a new concept to you. Basically, level of organization in the biological level start with the cell, and then the tissue, and then the organ, and then something else. We will discuss these levels of organization when we arrive at that area. Microscopes. Microscopes are tools for studying cells. Nowadays, many different types of microscopes are available for observing cells. Light microscopes and electron microscopes are two types of commonly used microscopes. The table below shows their differences. The image that is formed by light microscopes are basically formed by light rays. The basic working principle of light microscope is light passing through a specimen or passing through a thin slide of a specimen to form an image. For electron microscope, there are two types of electron microscopes. The first one is transmission electron microscope. Here, in transmission electron microscope, electron beam pass through a very thin slide of a specimen to form an image. Therefore, it can show internal structures of a specimen. Scanning electron microscope involve electron beams scanning over the surface of a specimen to form an image. Therefore, it shows external structures of a specimen. This electron microscope has a much larger magnification compared with light microscope, about 1 million times on average. For transmission electron microscope, mostly would be 1.5 million times. Most scanning electron microscope has a magnification of around 200,000 times. So, transmission electron microscope show internal structure and cannot form 3D images of a cell. Scanning electron microscope show external structures, therefore can form 3D images of a cell. In contrast of light microscope, electron microscope use electron beams instead of light rays to form images. The images displayed under the light microscope can be seen directly by the human eyes. However, for electron microscope, the image must be shown up on a fluorescent screen before it can be seen by us, the human eye. The image color of light microscope is colorful. But for electron microscope, it can only be black and white images because electron beam has no color. But sometimes you can see electron microscope having different colors on your textbook. That is because some computer graphics has been done to the photomicrograph to make it colorful, so that different structures can be distinctively shown. For the differences in resolution between light microscope and electron microscope, resolution means the details of the cell structure. Light microscope can show the structures inside cells as small as 2 μm. 1 μm equals 10 to the power minus 6 meter. 1 μm means 1 micrometer. So basically 1 micrometer equals 10 to the power minus 6 meter. At this level of resolution, structures such as nucleus, cell wall, cell membrane, cytoplasm, starch granules, and chloroplast can be identified, while other structures cannot. For electron microscope, the resolution can show the structures inside the cells as small as 1 nanometer. 1 nanometer is equivalent to 10 to the power minus 9 meter. Here, with an electron microscope, what can be seen by the light microscope can now be seen in greater details. Structures like the rough ER, smooth ER, that is the endoplasmic reticulum, ribosomes, and mitochondria can be seen in greater details. Now we will come to the part use of light microscope. The main parts of a light microscope that are used for magnifying the image are called the eyepiece and the objective. Both of them have lenses. Therefore, they can magnify the image. The main parts of a light microscope that are used for focusing would be called the fine adjustment knob as well as the coarse adjustment knob. When you are focusing on a specimen, you first turn the coarse adjustment knob to bring about a large movement of the stage to make a fast focusing first. After that, you use the fine adjustment knob to make a more accurate focus. What I've just said are the most important parts of the light microscope, responsible for magnifying the image and making an accurate focus. There are other important parts, such as the mirror or the lamp. Most probably the lamp will be used in the laboratory in nowadays science school science laboratory. The mirror or the lamp provide light through the specimen. Just below the stage of the microscope, you will see the condenser. Condenser is used to focus the light from the lamp to the specimen. The condenser has a lever to control the diameter of the hole of the diaphragm, so that's to control the amount of light passing through the specimen. That is, you can control the brightness by adjusting the diaphragm. Usually at high power magnification, the light passing through the field of view, that means the area of specimen being observed, will be less. Then you have to adjust the diaphragm so that more light rays can pass through the specimen to make the brightness increase. Please refer to chapter 2, page 16 of your textbook for more information about what I've just said. There are a number of special properties of the light microscope. Magnification. This word represents how many times an image is larger than the object. The overall magnification of a light microscope depends on the objective and the eyepiece used. It can be calculated by the overall magnification equals magnification of the eyepiece times the magnification of the objective. For example, if a 20x eyepiece and a 40x objective are used, the total magnification of the microscope would be 800x. That means 800 times larger than the original object or specimen. To view more cells under the microscope, you should use an objective lens of lower magnification. This is because at low power magnification, the area of specimen observed is larger than that of high power magnification. You can see that the high power magnification only enlarge the middle part of the low power magnification. The area being observed at high power magnification, the area of specimen being observed at high power magnification is smaller than that of low power magnification. Next, when switched from a low magnification to a higher magnification, the image will become dimmer. So imagine the green dots here are the light rays passing through the low power magnification's field of view. Imagine how many light rays will be passing through the high power magnification. You can only see here 1 and 2 light rays. You can think that as 2 light rays passing through the field of view of high power magnification. Therefore, it is dimmer. The image is dimmer. In this case, if you remember, what do you have to adjust? Yes, you have to adjust the diaphragm so that more light can pass through the diaphragm, and then so that the image of the high power magnification would be brighter. Now we will move to talk about cell theory. Due to the development of the microscope that we have just been talking about, the cell theory is proposed as below. There are 3 basic statements in the cell theory. Number 1, all organisms are made up of 1 or more cells. You can regard this as a fact. All cells come from pre-existing cells. You can regard this as another fact to remember, so that you can have logical thinking, kind of deduction later on when you are asked in examination questions. The third statement in cell theory states that the cell is the basic unit of life. It is the smallest unit, not just smallest unit. It is the smallest unit that shows all 7 characteristics of life. For example, nutrition, respiration, reproduction, excretion, to name just a few. The next area that we are going to talk about is, of course, the cell itself. There are hundreds of different types of cells in organisms. They vary, they change in shape and size. They are different in shape and size. But there are some similarities between them. Plant cells and animal cells, as shown here, generally share some basic structure. The major parts of both of them is a jelly-like fluid called cytoplasm. The cytoplasm is bounded by a cell membrane. So, cell membrane. Various organelles, for example, the nucleus, endoplasmic reticulum, Mitochondria. Vacuoles is a small vesicle, or called small vacuole. It's a big vacuole in plant. Ribosomes. If you can see here, some ribosomes. Also present in plant, ribosome. And chloroplast only present in plants. All these organelles are held in the cytoplasm. Now we go into different structures in detail. The nucleus is a spherical structure bounded by a double membrane called the nuclear membrane. I try to draw this out for you. This is a transverse section, TS, of the spherical nucleus. There are pores on the surface of the nuclear membrane, which is a double membrane. These pores allow exchange of materials between the nucleus and the cytoplasm. The nucleus contains DNA, the genetic material of a cell. DNA carries genetic information which controls the activities of the cell. The activities literally means the chemical reaction, doesn't mean some moving activity. No, it means chemical reaction. So, in short, nucleus enclosed by a double membrane perforated by nuclear pores. Its function is to synthesize and contain DNA. The rough ER, the rough endoplasmic reticulum, has the features of having many ribosomes attached on the outer surface. That is like this. You can see the dots, the ribosomes attached onto the surface of onto the outer surface. It has interconnected membraneless sac. Its function is for making protein, as well as transporting the protein made. The smooth endoplasmic reticulum has the feature of also interconnected membraneless sac. However, there are no attachment of ribosomes. Its function is for making lipids and transporting lipids. Mitochondria is a rod-shaped organelle bounded by double membrane. The outer membrane is moved while inner membrane is highly folded like this. The function of mitochondria is to carry out respiration, of course. Aerobic respiration means respiration that requires oxygen. It is also responsible, of course, for producing ATP because this is the product of respiration, which is the form of energy to drive cellular activities. Again, activity doesn't literally mean movement, but it means chemical reactions. So you make sure that you get this concept word correct. Chloroplast is responsible for photosynthesis. It has a system of flattened membrane-bound sacs to hold chlorophyll. Inside chloroplast, there is a system of flattened membrane-bound sac to hold chlorophyll, the name of which you will learn later. It has another feature of being oval in shape. and it's bounded by a double membrane. It carries out photosynthesis by converting light energy to chemical energy stored in carbohydrates, the glucose, for example. Vacuole, having the feature of a fluid-filled sac bounded by only one single membrane, unlike the chloroplast and mitochondria and the nucleus having double membrane. Vacuole has a single membrane only. Its function is for transport or food digestion. So number one, transport, and number two function, transport of materials, of substance, of things inside the cell, or food digestion in animal cells. The literal mechanism, the literal process, that means the actual process of which you will learn later on, but not now. You just kind of remember it first. Another function is to help keep plant cell, well, we say, in a turgid state. You kind of just remember this word first. More information of the concept of turgidity or turgid state will be learned in Form 4 in Chapter 3. The other function of the vacuole is for storage of water and food. Another function is for containing waste, so that these waste will not interfere, that means disturb the normal metabolism, means the chemical reactions in the cells. This is a pretty important word, so you learn right now, metabolism, chemical reactions in the cell. So these wastes are being compartmentalized, being confined inside the vacuole so that it doesn't interfere, doesn't go into the normal metabolic reaction, doesn't affect the normal metabolism of the cytoplasm. The cell membrane has the feature of being differentially permeable, as comparing with the function of cell wall, which is fully permeable, which means basically allow any substance to pass in and out freely. The cell membrane has the ability to select things that can pass into and out of the cell. We say differentially permeable. It is made up of protein and lipid. It controls the passage of substances into and out of the cell. The cell wall, in contrast, is not differentially permeable, it's fully permeable, allow things to go into and out freely. It is made up of cellulose. In Chinese, we say 纤维, and is therefore rigid, quite rigid. You can pull at your clothes, they are also made of cellulose. Function of cell wall would be to give shape, to give support, and to give protection. Number 1, 2, and 3. Please write it down yourself. Give shape. Most plant cells look like this, have a regular shape, and give support. It is harder, rigid, so it gives support. And because it is rigid, it gives protection to the plant cell too. Now we come to a comparison between plant cell and animal cell. There are some similarities. Both of them have cell membrane, cytoplasm, nucleus, and mitochondria generally. Some cells do not have nucleus, like the red blood cell, but we will talk about that a little bit later on. So These are the similarities. The differences would be plant cell has cell walls and animal cell have no cell walls. Plant cell have a large central vacuole while animal has no large vacuole. They may have small vacuole called vesicle. A plant cell have sometimes has chloroplast because they are confined to green cells only. For animal cells, strictly no chloroplast. I am going to introduce to you some cell types that you should, as a biological student, as a biology student, you should remember at least a number of animal cell types. The red blood cell. There are some information. Number one, it has no nucleus, as I have just talked about. It has the importance. It is biconcave in shape. This biconcave shape has the importance to provide a large surface area to volume ratio, so that to facilitate the uptake of oxygen. A large surface area to volume ratio help increase the efficiency of the uptake of oxygen into the red blood cell from outside, for example from the lungs. The other type of animal cell would be white blood cell, for example the phagocyte. This white blood cell has looped nucleus, and it can change shape like an amoeba to engulf bacteria. And white blood cell, there is another type of white blood cell, that is the lymphocyte. The lymphocyte has only one large oval nucleus. It has the function, it is responsible for killing bacteria or destroying. We don't say killing virus because virus such as the COVID-19 has no signs of life. So we say destroy virus instead of killing virus. You don't say kill. Destroy virus. They destroy them by producing antibodies. These antibodies are Y-shaped protein stuff, and they are responsible for killing bacteria and virus, and destroying or damaging the structure of virus. Another type of animal cell would be neuron. It is drawn like this. This is the nucleus, and these are the way how you represent the neuron. That means the nerve cell. They have long cellular extension or branched endings. They are responsible for transmitting nerve impulses, which are the electrical signals that is transmitting throughout our bodies. Another kind of animal cells are mucus secreting cells and ciliated cells, or what sometimes we call ciliated epithelial cells, means cells on the surface of the skin or tissue. But mucus secreting cell serves to secrete mucus, and these mucus trap dust particles. For example, they are present on the breathing tract. That means your trachea, to trap the dust in the incoming air. The ciliated cells has cilia to beat, to drive object. That means to drive the mucus together with the trapped dust along the breathing tract, so that they can be coughed out or swallowed into the stomach, so that they don't get into the lungs. Sperm cells, I'm sure you are very familiar with this. It has the head containing the nucleus for fusion, that means for mixing up with the female nucleus, and it has the tail for swimming. The egg cell has large cytoplasm. These large cytoplasm are required to store nutrients. Plant cells. The first type of plant cell I would like to introduce you are the root hair cell. The hair sticking out of the cell can provide that means the root hair can provide a large surface area for water absorption and salt absorption, and also it can assist gas exchange. Xylem vessel. Xylem vessel are cells that are present in the veins of the leaves, as well as what we call the vascular bundles, which you have learned in Form 1 inside the stem or the leaves of the plant. They are dead cells. They don't have nucleus, they don't have cytoplasm, they just have cell wall. They are hollow tubes. They help transport water. They have hard, thickened cell wall, even thicker than usual cell wall. They are hardened, they are thickened by a certain chemical called lignin that you will learn later on in Form 4. And because they are hard and thickened, they provide mechanical support so that when water is being sucked up the plant, the negative pressure created may cause collapse of the xylem vessel. But when the xylem vessel is thick and hardened, this negative pressure created when the water goes up, the thick and hardened cell wall literally prevent it from collapsing. Another type of plant cell would be the thin-walled cells. Very thin. It can be stretched. That means it can be stretched to a certain extent. So it has some information here. It has thin cell wall and large central vacuole. It stores water and nutrients. It supports the plant by cell turgidity. That means the cell becoming bigger and bigger until it cannot become any more bigger. Then we say that the cell is in a stage of being turgid, or is in a state of turgidity. So you don't have to wait until Form 4 to understand this word, because I have just said it to you. But you can just type into the Google and find a little bit more about this word yourself for interest's sake. Guard cells. Guard cells has a thicker inner wall, has a thicker inner cell wall, and a thin outer cell wall. Thin outer cell wall. A thick inner wall and a thin outer cell wall. It has a special reason why the guard cell is like this. It is adapting to certain function. And these certain functions help to control first by controlling the size of the stoma. That means the hole inside the two guard cells, stoma. Of course, these are just some brief information, more information of which you will learn in Form 4. It is sausage shape, at least you should remember this, and you should also remember that it possesses chloroplast. It is green in color. Lastly, I would like to introduce you to onion's epidermal cell. That means the surface cell, the outermost cell of the onion. rather say the surface of the surface cell of the onion tissue. It possesses no chloroplast, as you have already seen in Form 1. When you look at the microscope, you will see onion epidermal cell. It has cuticle on the outer surface to reduce water loss and also to prevent entry of pathogens. So one, to reduce water loss, the cuticle is what we are called a waxy layer. And this waxy layer can, of course, you can understand why it is waxy, so it can reduce water loss. It also can serve to prevent entry of pathogens. Pathogens means disease-causing agents. For example, viruses such as the coronavirus, for example, the COVID-19 that we are experiencing now, which is this virus. This is another type of virus, not the coronavirus. So pathogens are disease-causing agents. Lastly, I would like to talk about the level of organization of living things. The basic level, the most basic level would be cell. The second level would be tissue, which is defined as similar cells grouped together to form tissue. For example, muscular tissue, bone tissue, cartilage, 軟骨組織 cartilage, blood is also a kind of tissue because by definition, this is the definition, blood is a group of similar cells, red blood cells, going together to serve similar function. Plant tissue, examples of which are xylem tissue consisting of xylem cells, phloem tissue consisting of phloem cells, and companion cells, but we will learn about that later on. The mesophyll cell, the meso means the middle, phyll means leaf, that means the middle of the leaf, the mesophyll cells, 葉肉細胞, the epidermis, 表皮細胞, on the surface of the leaf. Organ has the definition of different tissue grouping together to carry out specialized function. For example, the stomach of an animal organ, small intestine, liver, pancreas, kidney, lung, heart, brain, testis, ovary, eye, ear, and skin are examples of animal organs. And particularly, you might focus on to try to remember, but it's a little bit more difficult to remember the plant organs because you are less familiar with it. By definition, different tissue going together. The roots have different tissue going together. The stem has different tissue going together. Therefore, by definition, they are called organs. Leaves, flowers, buds, etc. As for systems are formed by grouping tissue and organs together. For example, digestive system consists of the stomach, alimentary canal, and pancreas, and to name just a few. The nervous system consists of your brain, of your spinal cord. Endocrine system consists of your glands. Reproductive system consists of your uterus, of the testes. The blood circulatory system consists of the heart, the blood vessels, and the blood itself. So it all comes together, the tissues and organs, to become system. And of course, after system, a different system going together would be this is the third, this is the fourth level, and the fifth level would obviously be the organism itself. That means the living thing itself. This is basically what I would like to say. In your book, the part 2.1 Chemicals of Life and 2.2 4B are not discussed here because they will be referred back to when you go up to Form 4. So this is what basically I would like you to know in Form 3 basically. That is some information about the microscope, some information about the cell, and some information about the level of organization of living things.